Hey, Mr. P here. Oh, y'all are too much. Here we are for another exciting edition of Chapter 10. This is the second lecture in the 4-lecture series that is Chapter 10. In this video, we're going to talk about the differences between eukaryotic and prokaryotic cell division. Specifically, that eukaryotic cell division involves mitosis, and prokaryotic cell division involves binary fission. These are basically the same processes, but due to the structural differences of eukaryotes and prokaryotes, there has to be some process differences in how they divide their chromosomes. But both of them are going to involve cytokinesis. And remember in the last lecture I talked about what cytokinesis is and how mitosis and cell division differ. Cytokinesis, remember, is more synonymous with cell division because in cytokinesis, which literally means the movement of the cell, meaning we're going to divide the cell by moving two sides of that cell to opposite sides. And so we're splitting the cytoplasm. That is cytokinesis. And so not only are we going to talk about the differences between eukaryotes and prokaryotes, we're also going to talk about the structure of DNA and how that structure of DNA, whether it's chromatin, chromosomes, or chromatid, how that structure is going to really dictate how this cell division is going to happen. And so let's talk a little bit about prokaryotic cell division. As in previous chapters, specifically Chapter 7, we talked about what prokaryotic cell means, or what the word prokaryote means. And prokaryote, remember, is the type of cell that doesn't contain a nucleus. Okay, these are cells that are more ancient in history, okay, or more ancient in structure. And so these cells, because they're more ancient and because they're more simplistic, they're going to go through a reproduction that is asexual. Asexual means that it is not sexual. Okay, not sexual. Sexual reproduction, remember, is the combination, or it requires a combination of sperm cell, egg cell, fusing to form a zygote in order to produce a new individual of a particular species. This is the opposite of sexual reproduction, and so we call it asexual. Really the goal of binary fission is to take a single prokaryotic cell and essentially duplicate it, produce two identical cells that are genetically identical to that original parent cell. And so we call that binary fission. And binary fission is literally dividing that cell in half. Bacteria specifically, or prokaryotes, have a single, basically a single chromosome. And the chromosome is usually in the form of a plasmid, or they have plasmids and also contain a single circular chromosome. And so just because they contain their DNA in a nucleoid region and not within a nucleus, they still need to divide that nucleus content, or that nuclear content, meaning they need to divide their single chromosome in order to divide the cell and give each new cell a copy of that DNA. Remember, every new daughter cell needs to contain essentially a copy of each original parent's DNA in order for that cell to be genetically identical to that original cell. And so that bacterial DNA needs to replicate first, then it needs to separate, and then the cell can divide using that cytokinesis process. Okay, that cell divides into two pieces. Talking specifically about plasmid, bacteria have this process called conjugation that will allow them to kind of pass on their inheritance via this plasmid, and that's called conjugation, and it takes a conjugation, or requires a conjugation pili, but they essentially inject this extra chromosomal material into an adjacent bacterial cell in order to kind of influence the way that that next or adjacent chromosome or bacteria cell is going to behave or act. That is not a form of sexual reproduction. That is just a kind of a secondary reproductive process that allows bacteria to kind of differentiate themselves and helps with this whole idea of natural selection and evolution. And so if we look at an image of binary fission, you'll see that one single prokaryotic cell begins this entire process. Again, the chromosome is contained within a nucleoid region. There is no membrane around that region, and so it lacks a true nucleus. That's where we get the word prokaryotic. There is a cell membrane, there's a plasma membrane, a cell wall, and it has all the structures that we talked about in Chapter 7, but it doesn't have a nucleus, and so we don't need to go through the same mitotic division, meaning we don't need to go through prophase, metaphase, anaphase, or telophase in order to divide this cell. We are simply going to duplicate the chromosome, meaning we're going to copy the chromosome, and now we have 1, 2 copies of that DNA. Notice that there's twice as much genetic material here as there was originally. And, excuse me, as we duplicate this chromosome, it's kind of starting to move towards the poles a little bit, and it's not going to move a lot, but it's going to kind of separate itself, and then we're going to eventually divide the cytoplasm right along this kind of plane. And you'll see right here the membrane and the cell wall pinching in. This is obviously where that cell cytoplasm is going to divide, and eventually we will have 2 identical daughter cells that are genetically the exact same or genetically identical to our original parent cell. Okay, so essentially one cell becomes 2 by replicating its chromosome and then cutting itself in half. If we contrast that with eukaryotic cell division, it requires mitosis, and so that kind of, before we talk about mitosis, we got to talk about the states that DNA are in at specific times within a cell cycle. The 2 states of DNA that you will see in and around the cell cycle is chromatin and chromosomes. Chromatin is the long, thin, stringy state of DNA. It is the normal version of DNA, meaning when the cell is not dividing, the DNA is in the form of chromatin. And so I know we haven't talked about the cell cycle in detail yet, but interphase is basically the entire cell life cycle other than when it's dividing. And so when the cell is just naturally doing its cellular things, the DNA is going to always be in the state of chromatin. Now, chromatin is a fairly coiled, fairly structural form. It's not like it's not structured. It is structured, but chromosomes are a way in which we supercoil this DNA or condense it further. And in order to take chromatin and move it to a chromosome state, we have to wrap it around tiny proteins called histones. These tiny proteins are kind of like spools, and everybody kind of knows what like fishing line spooled around the spool looks like, or what thread spooled around a spool looks like. The reason long strands of material come on spools is because it's easier and more organized that way. Nobody goes and buys a handful of thread, because when you go to use it, it's all going to be tangled up and it's going to be really bad, and you're going to be in trebs, and then you're not going to be able to use it, and so it's going to be a mess. But it comes on a spool so that it comes off the spool nicely and doesn't get wrapped up. It's more compact and it's more organized that way. Okay, so we have chromatin and chromosome. Chromatin is the long stringy strands of DNA that are found during interphase. Chromosomes are the supercoiled, wrapped around histone proteins, more compact, more organized form of DNA, and those chromosomes are only found during M phase of mitosis. Okay, during M phase only. Now, let's talk about the chromosome structure. This is an actual micrograph of a chromosome. Now, this is a transmission electron microscope, okay, or micrograph, and so this is an actual image of a chromosome. You can see that if I outline the shape of this chromosome, it's an X, okay? And so when you see other drawings of chromosomes or actual animations of chromosomes, you typically see them in X form. There is a structure in the middle called a centromere that holds the 2 sister chromatids together. So there's several different vocab words we're going to talk about. We have chromatin, which I talked about before. Chromatin is the long stringy strands of DNA. You supercoil chromatin to form a chromosome, which is an X-shaped chromosome. Okay, it's an X-shaped structure. A chromosome is made up of two sister chromatids that are held together by a centromere, and that centromere is going to attach to a spindle fiber, which we'll talk about later, and will attach to a centriole when we start talking about mitosis and actually pulling these chromosomes to the middle of a cell and then eventually pulling these chromosomes apart. But for now, no chromatids, no chromatin, no centromere, no chromosome, and no centriole. Okay, centriole, C-E-N-T-R-I-A-L, centriole. Here is our DNA structure. This is the most unstructured, the very most kind of simplistic version of DNA is just the DNA double helix. Okay, I know everybody's seen this. It's like the twisted ladder. There are two strands. You have one strand here, one strand here. It's like a ribbon that you twist. That is the DNA double helix. Well, if we zoom out, this blue strand here is this blue strand here. So we've zoomed way out, and we're going to wrap that DNA, this long double helix, around histone proteins. Okay, and these histone proteins are basically in these octet or these structures of multiple histones. And so we're going to wrap them around the histones like thread on a spool, and then we're going to even zoom out further, and we're going to coil this strand around itself, and then we're going to supercoil DNA, and we're going to take this strand and we're going to coil that strand. And so you can see from here to here to here to here, we're getting more complex as we go. Supercoiled DNA is the version of DNA that chromosomes are formed from. Chromatin, you can see how much more complex it is than even just the DNA double helix, but again, it's not the most complex form of chromosome or DNA out there. So, talk a little bit about those states of DNA. I just showed you a picture of them. Now we have some text. Remember, chromosomes are one of the two states. We have chromatin, which is the kind of the usual state of DNA. Then we have chromosomes, which is the version of DNA that is found during DNA replication, okay, or during cell division. And when you take the chromatin and form the chromosome, if the chromosome is replicated, it is an X-shaped. I showed you what that X-shaped micrograph looked like. Each arm of the X is a sister chromatid. Remember, you have one sister chromatid, looks like this. You have another sister chromatid that is attached to the same middle point. That same, this middle point right here is called a centromere. And the centromere is the structure that attaches the two sister chromatids to each other. Okay, this is one sister chromatid. This is the other sister chromatid. Two sister chromatids make up one X, which make up one chromosome. Okay, this copy should be identical to this copy. So if this has a gene on it that is a dominant A, this one should have a gene on it that is a dominant A. And if this arm or chromatid has a dominant B, this chromatid should have a dominant B. Now there are going to be some differences when we talk about homologous chromosomes, but that's going to be later on when we talk about genetics. But for now, as long as you know that chromatids are held together by a centromere and that chromatids contain genes that are identical to each other, really that should be all you need to know at this point. Exact duplication ensures each new cell is identical genetically because we want to make this chromatid from this chromatid so that when we split this chromosome in half and this chromatid goes to that cell and this chromatid goes to this cell, each new cell is identical, meaning this cell over here has a big A gene and a big B gene, and this one over here has a big A gene or a big A allele, and this one has a big B allele. Both cells are identical. Okay, that's the purpose of mitosis. Ensure genetic continuity. And the last thing we're going to talk about is two types of cells in the human body. The entire body, is made up of two types of cells. There is a large majority of cells in this category called somatic cells. Another name of somatic cells is body cells, and these are the cells that make up the structure that is you. Okay, so all of your bone cells and all of your nerve cells and all of your muscle cells and skin cells and blood cells and brain cells and organ cells and eyeball cells and all that stuff. All of the cells that make up the body are called body cells, and the fancy word that you need to know is somatic cells. The only other type of cell you have in your body, and you don't have both of these, but you have one type, is the sex cells. Males are going to have males are going to have sperm cells, and if I can erase this, males don't have eggs. Males have sperm cells, and females have eggs. Okay, so a female is made up of all of these somatic cells that make up the body and egg cells. Egg cells are the sex cells. We call these gametes. Okay, gametes. Gametes. Eggs are gametes. Sperm cells are gametes. Sperm cells are found in males. Egg cells are found in females. Both males and females have somatic cells. Okay, and so I think that's going to do it for this lecture. Nope, one more thing. Somatic cells. So a little bit more information about somatic cells. If I can erase this out of my way. Somatic cells are your body cells, and they are diploid. Okay, and diploid is represented by the symbol 2N, and what that means is N is the number of chromosomes in a haploid cell, and so diploid, because they produce pairs of chromosomes, we say they have 2 times the number of chromosomes in a haploid cell. Okay, haploid meaning half the total number of chromosomes are sex cells. So sex cells are haploid, somatic cells are diploid, and that makes total sense. Humans need to have 46 chromosomes. 46 chromosomes are what make humans humans. Not all organisms contain 46 chromosomes. Fruit flies, I believe, have 4 chromosomes. Dogs have, I think, more chromosomes. Cats have fewer chromosomes than dogs. I'd have to look up the exact numbers, but different organisms have different numbers of chromosomes. We have 46 chromosomes. That means that each of the gametes, each of the sex cells, have to have half that number, 23, because when you pair a sperm cell with 23 with an egg cell that has 23, get a cell with 46 total chromosomes. If we were to look at a sperm cell that had 46 chromosomes and an egg cell that had 46 chromosomes and bound those together, then you would end up with a cell that had 92 chromosomes in it, and that's obviously double the number we need to be a human. And so these 23 chromosome sex cells are, like you said, they only have 23 chromosomes, and so we consider that haploid. Somatic cells would be all other types of cells, and we call those diploid because they contain 23 pairs, or a total number of 46 chromosomes. Okay, so I think that's where we're going to leave it. That's it. Bye.